Hi everyone, I am Tommy. I'm a human nutrition student at the university, and today I'm going to explain to you the glucose metabolism so you can make it clear and understand it well so you can achieve good marks on your test. Okay, so let's begin. It always starts with glucose outside the cell. Glucose uses a glucose transporter to enter the cell. In the liver, we have the GLUT2 transporter, which is not insulin dependent, and on the muscle, for example, we have the glucose transporter number 4, which is insulin dependent. Okay, so once we have the glucose inside the cell, the exokinase in the muscle or the glucokinase in the liver will act on the glucose, converting it to glucose 6-phosphate. Exo means 6, kinase means to add a phosphate. That's what this enzyme does, is adding phosphate on which carbon? The carbon number 6. It requires ATP since this is the source of the phosphate group. Okay, so you can see here there is a green point and a red one, because this is the regulation. Allosteric regulation of this enzyme is due to high quantities of ATP stimulates this enzyme, and high quantities of glucose 6-phosphate actually inhibit this enzyme. Okay, so now we are going to glycolysis. Glucose 6-phosphate is converted to fructose 6-phosphate by the use of an isomerase, phosphoglucose isomerase. Fructose 6-phosphate is then converted into fructose 1,6-bisphosphate. So we have another kinase here, the phosphofructokinase 1, that requires ATP for the phosphate group again. And where is this phosphate group going to go? To the carbon number 1. Okay, now we have a 6-carbon molecule with 2 phosphate groups. We're going to split it in half by the use of an aldolase. This enzyme is an aldolase that creates DHAP, which is dihydroxyacetone phosphate, and glyceraldehyde 3-phosphate. But why is there here a 2? Because the hydroxyacetone phosphate, to continue the glycolysis, is going to get converted into glyceraldehyde 3-phosphate. Now if we continue, we have the conversion of glyceraldehyde 3-phosphate into 1,3-bisphosphoglycerate. This process is modulated by an enzyme called the glyceraldehyde 3-phosphate dehydrogenase. What it does is it gives the hydrogen from the carbon 1 to the NAD positive to make NADH. And at the same time, it gets a phosphate and replaces the hydrogen on the carbon number 1. So we get 1,3-bisphosphoglycerate. If we continue, we have the conversion of 1,3-bisphosphoglycerate into 3-phosphoglycerate. So we get rid of the phosphate we added recently. And this is funny because what is the point of doing this? Well, to create ATP. By doing that, we can create ATP, which is energy. And then from 3-phosphoglycerate, we get to 2-phosphoglycerate. That's a mutase. It simply gets the phosphate from the carbon 3 and it puts it on the carbon number 2. Then from 2-phosphoglycerate, we get to phosphoenol pyruvate. Phosphoenol pyruvate is then converted into pyruvate, which is the final step of glycolysis, the final product, via the pyruvate kinase, an enzyme that also creates ATP. So as we can see here, this is the whole structure of glycolysis, and as you can see, there are some arrows that are bigger, and the The name of the arrow is in bold because these enzymes are not reversible. Hexokinase is not reversible. Phosphofructokinase 1 is not reversible. And pyruvate kinase is not reversible. So, if we want to make gluconeogenesis in the liver, we can because all the arrows are reversible. But to avoid the use of these enzymes, which is not reversible, we need to make use of all the enzymes. As we can see here, to create glucose de novo, we have to use from pyruvate the pyruvate carboxylates. What it does is adding a carbon to the pyruvate via the CO2 requiring ATP. This is a free carbon molecule. Oxaloacetate, which is a metabolite of the Krebs cycle, is a 4 carbon molecule. Then oxaloacetate is going to get converted back into phosphoenol pyruvate by the phosphoenol pyruvate carboxykinase. This word means that the carbon is replaced by a phosphate group because kinase means phosphate. So as you can see here, GTP is used in this case to get the phosphate, and you get rid of the carbon. Phosphoenol pyruvate is then the molecule with 3 carbons and a phosphate group. These steps are all reversible until we get here to fructose 1,6-bis phosphate. We are going to need the fructose 1,6-bis phosphatase. Phosphatase enzymes, they get the phosphate group from the molecules. As you can see here, we have 2 phosphate groups. Basically, this enzyme is getting the phosphate on the carbon number 1. So we can get back to fructose 6-phosphate. This step is reversible, and then here, to create glucose from glucose 6-phosphate, we are going to use another phosphatase, the glucose 6-phosphatase, to get rid of the phosphate on the carbon number 6. And here we have glucose de novo. The regulation of all of this is via allosteric, as I said earlier, in the first enzyme, and hormonal regulation is also implicated. As you can see here from the arrows, we have glucagon, which is stimulating gluconeogenesis in the liver, and insulin, which is stimulating glycolysis in the muscles, for example. Here we have pyruvate. Pyruvate, depending on the circumstances, can be converted into many things. If there is oxygen, it can be converted into acetyl-CoA via the pyruvate dehydrogenase complex, which we are going to view in another video, because these then will enter the Krebs cycle. If there is not oxygen, pyruvate is going to get fermentation, and it can be ethanolic fermentation to create ethanol, or lactic fermentation to create lactate. And lactate is very interesting because it can go to the bloodstream and then eventually in the liver, and in the liver lactate can be converted back into pyruvate that can be converted back into glucose via gluconeogenesis. I hope this video will help you, and let's see you on the next one.